Assalamu alaikum dear students, welcome back to my YouTube channel. In this video we will discuss DNA replication MCQs part 3. Which process follows DNA replication? Option A, cell division, B, transcription, C, translation. Correct option is A, cell division. Where does DNA replication begin? Option A, centromere, B, telomere, C, origin of replication, D, promoter. Correct option is C, origin of replication. Which enzyme removes incorrectly paired nucleotides? Option A, primase, B, helicase, C, DNA polymerase, D, ligase. Correct option is C, DNA polymerase. Subscribe now and press the bell icon. Never miss an update from my technologist. Which term describes the old DNA strand and replication? Option A, daughter strand, B, lagging strand, C, parent strand, D, Okazaki strand. Correct option is C, parent strand. What stabilizes the replication fork? Option A, primase, B, ligase, C, single strand binding protein, D, telomerase. Correct option is C, single strand binding protein. Which enzyme prevents premature DNA shortening? Option A, DNA polymerase, B, telomerase, C, helicase, D, ligase. Correct option is B, telomerase. Which enzyme catalyzes phosphodiester bond formation between Okazaki fragments? Option A, helicase, B, ligase, C, DNA polymerase, D, primase. Correct option is B, ligase. Which enzyme prevents premature DNA shortening? Option A, DNA polymerase, B, telomerase, C, helicase, option D, ligase. Correct option is B, telomerase. Which enzyme removes incorrectly paired nucleotides? Option A, primase, B, helicase, C, DNA polymerase, D, ligase. Correct option is C, DNA polymerase. Which term describes the old DNA strand and replication? Option A, daughter strand, B, lagging strand, C, parent strand, D, Okazaki strand. Correct option is C, parent strand. Which strand is synthesized toward the replication fork? Option A, leading strand, B, lagging strand, C, both strands, D, neither strand. Correct option is A, leading strand. Where does DNA replication begin? Option A, telomerase, B, telomere, C, origin of replication, D, promoter. Correct option is C, origin of replication. Which protein prevents separated DNA strands from re-annealing? Correct option is option A, DNA ligase, B, primase, C, single strand binding protein, D, helicase. Correct option is C, single strand binding protein. What would happen if DNA ligase were non-functional? Option A, replication would stop. B, Okazaki fragment would not be joined. C, DNA unwinding would fail. D, proofreading would be impaired. Correct option is B, Okazaki fragments would not be joined. How many origins of replication are present in prokaryotic DNA? Option A, 1, B, 2, C, multiple, D, none. Correct option is A, 1. Which enzyme removes positive supercoils ahead of the replication fork? Option A, helicase, B, ligase, C, topoisomerase, D, DNA polymerase. Correct option is C, topoisomerase. Which enzyme seals nicks in the DNA backbone? Option A, ligase, B, helicase, C, primase, D, DNA polymerase. Correct option is A, ligase. Which molecule acts as a primer for DNA replication? Option A, DNA, B, RNA, C, protein, option D, DNTP. Correct option is B, RNA. What type of replication occur in prokaryotes? Option A, linear replication, B, rolling circle replication, C, multiple origin replication, D, none of the above. Correct option is B, rolling circle replication. Which type of mutation occurs if DNA polymerase makes an error during replication? Option A, frame shift mutation, B, point mutation, C, silent mutation, D, insertion mutation. Correct option is B, point mutation. Which structure on a chromosome shortens with each round of the replication? Option A, centromere, B, telomere, C, origin of replication, D, histone. Correct option is B, telomere. What would happen if helicase was inhibited? Option A, DNA replication would stop. B, Okazaki fragments would disappear. C, RNA primers would not form. D, supercoiling would increase. Correct option is A, DNA replication would stop. Which component provides the energy for adding nucleotides and DNA replication? Option A, ATP, B, DNTP, C, GTP, D, AMP. Correct option is B, DNTP. Which enzyme synthesizes primer in eukaryotes? Option A, DNA polymerase 3, B, DNA polymerase alpha, C, DNA polymerase 1, option D, DNA ligase. Correct option is B, DNA polymerase alpha. Which enzyme is primarily responsible for removing RNA primers and replacing them with DNA in prokaryotic cells? Option A, DNA polymerase 3, B, DNA ligase, C, DNA polymerase 1, D, primase. Correct option is C, DNA polymerase 1. Which nucleotide is always the first in an RNA primer? Adenine, B, cytosine, C, uracil, D, guanine. Correct option is A, adenine. Which type of DNA replication occurs in bacteria? Option A, linear, B, rolling circle, C, semi-conservative, D, conservative. Correct option is C, semi-conservative. Which type of bond does DNA ligase form? Option A, hydrogen bond, B, phosphodiester bond, C, glycosidic bond, D, peptide bond. Correct option is B, phosphodiester bond. Which structure protects chromosome ends from degradation? Option A, centromere, B, telomere, C, origin of replication, D, exon. Correct option is B, telomere. Which enzyme removes RNA primers in eukaryotic cells? Option A, DNA polymerase 1, B, RNase H, C, ligase, D, helicase. Correct option is B, RNase H. Thank you for watching my videos.